we're going to talk a little bit about research strategy, and Sue is going to just outline how the LAM Foundation fund research and just highlight some of the actually amazing work they've done and the considerable impact they've made on LAM research through their funding streams. I'm just going to talk a little bit about some sort of blue sky sort of stuff about a new angle of treatment for LAM, just based on a couple of papers that have come out in the last year or so, one of them funded by the LAM Foundation. And then Debbie Clements is going to talk about some of the work she's been doing that's funded by LAM Action in our lab in Nottingham. So I'm going to hand over to Sue first of all. And take it away.
So I have just a few comments related to research. As you can imagine, when Sue Burns in 1993 heard a diagnosis for her then 23-year-old daughter, Andrea, that she had something called lymphangioliomyomatosis, she said, well, what are we going to do about it? And the doctors said, well, there's really nothing that can be done. And she says, well, what does the research say? She says, well, there really isn't any research. And Sue was a grade school music teacher, and she decided that those answers were not sufficient for her family or her daughter, and so she, not knowing, half as much as what we all know today, just took it upon herself to make a difference. And from the very beginning, research was the goal. And there was no money, and she started with letter writing campaigns and reaching out to her teachers union and calling her friends and doing all the volunteer stuff. And I know, having talked with Sue, that the first time they had enough money to write a check to a scientist was one of the most important days of her life, because she was just the person on the phone. There was really very little money coming in, and she was only talking to women with this diagnosis who were suffering. And so much has changed since then, and I wanted to, our main priority still is funding research in addition to the patient support. And I think in this new era, in our third decade now of doing this, the lines between patient support and research are really blurring because it's all now starting to overlap. So we have basic and bench research. When we see the pictures of the cells and the staining and the crazy labels and the, oh my gosh, am I really going to be able to follow all of these arrows? And the scientists at the bench, we fund a lot of basic research. Then there's moving on from those theories into what, this is totally a layman's interpretation of this, so please feel free to correct me if my words are incorrect, but preclinical, can we recreate this disease in animals and then try things and see what responds? There's a whole range of that. Clinical research, we've talked about, is when we have enough of a theory to think that something might work and we want to study it in people. So that was the Miles trial, that famous study that really led to the conclusion that if you take sirolimus, you steady out. If you stop taking it, it goes down. And there were 90 women who volunteered to be a part of that study. And keep in mind that half of them got a placebo and half of them got the drug. Nobody knows if it's going to work. It's a sacrifice and a gift that half of those women, because it worked, gave up part of their lung function for all of you. And the other half proved that it works. And so, again, when we go back to this gift of giving, being a part of research, being a part of an inhaled rapid trial, if you qualify and it feels good for you, can be another type of gift. But our scientists like to give these clinical research trials great names. Midas, mild, cola results, LAM, and whoops, LAM was, I think that was a typo. There was another study, but they all stand for something. So in the United States, mild is our study of studying earlier use of sirolimus at lower doses, which is what Professor Johnson was talking about earlier, is what if you have higher lung function and we give you a smaller dose at an earlier stage, will you lose less lung capacity? It's remarkable that a small disease like LAM has currently 7 open enrolling trials in the United States right now, and then there's more around the world. So this is great progress. I don't know what this is really called, but when we go back and look at data that's been collected over time, whether it's at the National Institutes of Health or digging into your database at the National Health Service and drawing conclusions from that. There are so many papers that come out because they're still writing new publications from the Miles trial data. It's just there's a multitude of data points in there that you can draw new interpretations from. The qualitative questionnaires, what's going on with quality of life, asking questions of patients and understanding what is the clinical experience. There's so many things we can study. And then I think there's the holy grail of linking all of these things together. That was an aha moment for me yesterday when I was talking with the LAM research team here, is they have access to the clinical data for people seen at the National Health Service clinic, and they can tie that to the basic research and the other research and draw even more conclusions. We don't necessarily have that in the United States because people are seen all over the place. So I asked Roya about, she just gave a very complex and brilliant presentation about what she was studying, and I said, well, what's next? What's your vision of where you want this to go? And she said, well, I want to tie what I'm seeing in the lab to what's happening in the real world, in the clinical experience of the patient. I was like, we all want that, but that was what was going to make this also perfect in coming up with real conclusions. So we at the LAM Foundation, I wanted to talk about, I'm going to do this really fast. This is an old slide from Dr. McCormick. Okay, so what does that mean in reality? If you start up on the left-hand side, getting patients organized. So in our world, that was Sue Burns in 1994 when she found the first 8 patients in the United States that she knew had LAM. And she grew that to the now over 2,200. But finding the patients, telling the story and engaging Dr. McCormick and his colleagues, starting to raise some money, led to the discoveries of understanding the disease, what we were talking about earlier with the mTOR pathway, the gene, the protein, the pathway, getting the national institutes involved, and then having enough data to design a trial. Well, you have to have the patients. So if Sue hadn't been working over on this side doing these kinds of meetings, we wouldn't have been ready to do anything with the funding, and then that led down to finding the target and doing all of those different types of research to ultimately find sirolimus. So that's how it actually happened. Sounds really simple on one slide, took 20 years, but that's the initiative that starts with the patients organizing. This is just a map of what was talked about earlier with the Miles trial. So these are the actual locations and the number of patients and where they lived who participated in the Miles trial that proved the effectiveness of sirolimus. So you had 2 sites in Japan, Toronto, and the other sites in the United States. And I think over the course of those years, the LAM Foundation donated about half of its net value to keeping this study open and going. And that was the actual publication in the New England Journal of Medicine. We have it framed in our offices. I hope you do too. So all of those kinds of research we've been funding, and this is very tiny, 1996, all the way up to 2017. The gray bars are scientists writing proposals, sending them to the LAM Foundation and saying, I think I have a great idea about what needs to happen with this aspect of LAM. Will you fund us? So those distributions are just shy of about $10 million that we've actually get the proposals in, have the scientists read it, look for the best science, and we're funding scientists in Japan, in Canada, in the United States, and this little place locally called Nottingham. They're doing some pretty good work there. And then in addition, so that's money donated to the foundation that then is just sent back out. The blue is sort of a technicality that I have to present in the United States. It's called the MDBR, which is the $1 Million Bike Ride, sponsored by the University of Pennsylvania. If you're a rare disease group and you form a cycling team and you raise $50,000, they will match it for a total of $100,000 that will go to your disease research. But it's a whole separate sort of, we write the check to them and then they write the check out. But we've raised this year we will be over $500,000 by having our disease, our LAM Foundation Easy Breathers ride 5 years in a row and raise the money that was then funded. So that's that, and that all goes to LAM research. And then in addition to that, all of the clinical, different funding for clinical trials, scientific meetings, and research projects. So that's where we get to the $14 million. Again, 90% of that is all raised by people just like you. Beer and wine tastings, 5Ks, bake sales, one-armed bake sales. And making a difference and keeping the scientists going. So the last one thing I want to share is a quote from Dr. McCormick, who really got all of this going. He said this at our gala, and it is his estimation that, in truth, the expertise that is assembled to study LAM and tuberous sclerosis, because they're linked genetically, could not be duplicated in terms of the scientific caliber around the world, even if you were a for-profit pharma or device company that had hundreds of millions of dollars. He said it just can't be done. So we are truly a fortunate group of people to have this caliber of scientists working for us. And they will get it solved. I do believe that. That's what I have to share about research and the importance of it and the importance of you staying involved in it. Thanks.
So that's a really great achievement over all that time by very many people. So I'm going to change gear completely. This is a sort of bit of a, as I said, a bit of a blue sky thinking sort of talk about potential future treatments for LAM. And I don't know if people have often thought, if anyone's immune to LAM, perhaps the question really should be why people aren't immune to LAM. And we have an immune system, and its job is to detect when things go wrong. And that's often infections, but actually if people get tumors or other significant chronic diseases, the immune system generally reacts to those things to try and oppose the change. And using the immune system to treat diseases is an expanding area, which I'll just highlight very briefly. So just to remind you, I showed you this slide this morning about what happens in LAM. The LAM cells acquire these mutations, mTOR's activated, they proliferate, they spread to the lungs, and in the lungs, the growing LAM cells damage the lung tissue by some mechanism, and the symptoms ensue. When we drew this slide, it's about 8 years old now for a review paper. What we didn't put in at all, and what there wasn't really much interest in at that time, was the role of the immune system in LAM. And of course, your immune system should detect that you have LAM and do something. The fact that it doesn't suggests that maybe that the LAM cells are putting up some sort of barrier to stop your immune system functioning normally. And this resistance to the immune system, or immune evasion, or there are various terms for it, is something that's commonly seen in a variety of cancers. And so tuning the immune system to attack cancer and other chronic diseases is a really growing area of medicine now. And I don't know how many people saw this lady in the news this week. So about 3 years ago, this lady was diagnosed with advanced breast cancer. She had metastases in many of her major organs and really was given only a couple of months of life expectancy at that time. She was fortunate to be accepted onto a novel treatment at the National Cancer Institute in the US. And what they did was they took her primary tumor and they took the metastatic tumors that had spread around her body. They analyzed them in the laboratory. They looked at everything that was different on the cell surface about her primary and her secondary tumors from the rest of her normal tissues. And then they took her immune cells and they trained her immune cells to react to those differences. They then expanded the immune cells and they fed back 90 billion activated immune cells into her body that was specifically tuned into her tumor. And these are the results. So this is a CT scan of her abdomen before treatment. And the arrows, those little round grey areas, they're all the size of a sort of golf ball probably, are all breast tumours that have spread to her liver. And so her liver's strongly activated by that. After the immune therapy, they have completely gone. And so this is now. This is her kayaking, and she spends her time kayaking and walking with her sort of new life that she's been given. 3 years later, she seems to have no evidence of breast cancer. And so this has obviously been an extremely exciting treatment and probably the best response that they could have expected. And isn't a routine thing, but this technology is being developed, and there's a lot of excitement for different diseases. So we're not at this stage with LAM clearly, but people are beginning to look at how the immune system and LAM might be modified to improve treatments. So what you need to know about the immune system, it comprises lots of cells, and some of them are cells that can be activated to kill tumors, to kill infected cells, to restore the system to normal. And then there are other cells that regulate this immune response. And one of your immune system's major tasks is determine what's foreign, what's diseased, from what's normal. And if it gets that wrong, then the body reacts against itself and you get autoimmune conditions like, for example, rheumatoid arthritis. So the immune system's kept very strongly in check. And the main effectors of the immune system are called T cells, and they can recognize and target abnormal cells using ligands called checkpoints. And the checkpoint is that when the immune system sees a cell, it just works out whether it needs to kill it or whether it needs to just pass on by. And modulating these checkpoints is an important therapy for cancer. This is a paper from Lisa Henske's group. Lisa's a tuberous sclerosis LAM researcher in Boston. That's the one in America, not Lincolnshire. And her group, active research group, and they asked the question of whether these checkpoints were important in LAM. So this little cartoon just shows the checkpoints, and you can see this tumor cell which is being recognized by the immune system, and the T cell at the bottom is the thing that usually kills T cells. And these checkpoint ligands tell the immune system actually to pass by and go, and this is a normal thing. And the tumor cells have now learned how to express these checkpoints and pretend that they're part of the normal tissues, and so not to be targeted by the immune system. So the first question to ask is, does this happen in LAM? And in fact, does the immune system recognize LAM at all? Well, the answer to that is yes. And the little figure up here is from the paper. The panel here is normal kidney, and the little brown dots are T cells that have been identified by an antibody. And you can see there's just a couple of T cells cruising around in a normal kidney, just looking for trouble, not finding much going on. This is an angiomyolipoma, a kidney tumor from a woman with LAM, and actually there are lots of T cells in there. You can see lots of brown dots. If you quantify this in lots of tumors, you can see that, yes, the immune system recognizes LAM and it recognizes angiomyolipoma. It doesn't seem to do much to it, though, really. The question they asked was, maybe if we block these checkpoints that perhaps the LAM cells are expressing, maybe we can affect the course of the disease. Now, we're not in a position to do this in people yet, but what the group did was they made tumors that look like LAM cells, and they put them into animals, and they looked at their growth. So I'm not going to say which color this is, but this line at the top here is a tumor growing in an animal that it grows quite quickly, as you can see. These lines here are when they use inhibitors of either one checkpoint called PD-1 or another called CTLA-4, and they put an antibody in that stops this working. What happens actually is that the immune system attacks the tumor and they grow much more slowly. They don't go away, but they actually grow more slowly, and one of these is a combination of both. And when they look at the tumors, what they see inside is that there are many more of the tumor-killing immune cells in the checkpoint inhibitor-treated tumors, and reduced numbers of the immune cells that sort of suppress the immune response. The next thing that they did was they looked at another model system where LAM or tuberous sclerosis-like tumors spontaneously form in animals, and they treated them with the checkpoint inhibitor therapy. And this is a number of new tumors that appear in the animals, and in the control group, you've got tumors forming relatively regularly. only about half as many in the immune-treated animals. So this is looking like you can potentially, at least in these model systems, reduce the growth of these cells that look like LAM cells. So these are initial steps towards these therapies. The other thing that they looked at, of course, is what is it about LAM cells that allows them to evade the immune response? And it sort of comes back to the mTOR pathway again, and the mTOR pathway is associated with the expression of these molecules. And what the group have done here is that they've used a combination of rapamycin and these new therapies in tumors. And they've treated the tumors for a little while with rapamycin, and with the blue ones, that they've then just stopped the therapy, and with the red line, they've used the checkpoint inhibitors, and this is actually significantly, together with the rapamycin, reduced the growth of these tumors. So these therapies are looking very promising. What's also good about these is these checkpoint inhibitor drugs are already used in cancer, and they're really quite helpful in cancers like malignant melanoma, and perhaps in the future may have an application for LAM. So that's a whole group of drugs that, again, we may be able to repurpose for LAM. This paper is on a similar theme about the immune response to LAM and how LAM avoids it. This group was funded by the LAM Foundation. This publication was in the Journal of Clinical Investigation Insight, which is a high-level science journal, and really looked at how natural killer cells, which are more of the business end of the immune system, and how they might target LAM. And usually, stressed or abnormal cells express molecules, as I've suggested, that the immune system activate, and these are called NKG2D ligands. And these aren't present on normal cells at all. It's slightly different from the checkpoint inhibitor situation. And the immune system recognizes these cells. So this is a biopsy of some lamb lungs, and these are two of these particular types of molecules that the immune system recognizes. These are stained with an antibody that fluoresces green. This red antibody identifies the LAM cells, and if the LAM cells express these proteins, then they go yellow in this slide. And you can see these proteins are strongly expressed in LAM nodules, so really should be attacked by these natural killer cells. The story's a little bit more complicated, and the reason why these cells don't attack them is because these ligands also get released into the bloodstream. Something cuts them off the surface of the LAM cells, and they float around in the blood. And this blocks the natural killer cells from recognizing them quite so well. And if you look at the levels of these cleaved proteins in the blood, and you compare them with what happens to women with LAM, you see a very interesting and important relationship. This is a slightly complicated graph, but these, well, actually, let's look at these shaded ones. These bars are how rapidly your lung function declines. If they can't detect these ligands in the bloodstream, these proteins, actually the disease is fairly quiescent. Lose a little bit of lung function, not very much. If you've got either one of these ligands, the disease is more active. If you've got both, these are the people who have the most rapid progression. And what the team thought was happening was that these ligands float off, and they act as little decoys for the cells that should be killing the tumors. And the cells are so busy mopping up these ligands in the bloodstream that they're unable to fight the tumors in the tissue. And of course, if we could find what sheds these molecules from the LAM cells into the bloodstream, and we could block that, we could then actually get the natural killer cells to target the LAM cells in situ. So we could train the immune system or modulate it to fight the disease for us, and hopefully turn this into this situation, which would obviously be of benefit. So that's really all I want to say. I think this is an important area of medicine generally for cancer, and the fact that people are looking at this in LAM just sort of tells us a little bit about LAM research, really. I mean, people are looking at all sorts of areas to try and counter this disease in different ways. And yes, we know about rapamycin now. They're looking for treatments to add on to rapamycin to make it more effective in the trials that Sue highlighted. And then there are all sorts of new areas that are being investigated, including some powerful new and rapidly moving areas. So this is a watch this space. So back to the UK, and I'm just going to hand over to Debbie Clements now, who's a postdoctoral researcher in our lab, who's going to tell us about some of the work she's doing that's funded by LAM Action. Debbie.
Simon said, I'm a postdoctoral, I'm a bench researcher, so I'm not a clinician at all. Yes, it is all fine, okay. So what I'm sort of going to talk about today is the role of one particular cell in LAM, and it's a very small part of the research that we do. But what I really want to talk about is how we use patient samples in our research. So actually how patient samples, they direct the type of research we do, they direct the questions that we ask, and they enable us to perform our research as well. So back to school a little bit. This is the very tiniest bit of the lung, the tiniest functional bit of the lung. There's an airway coming down, and these balloon-like things are the alveoli. Now, if you've had a biopsy or a lung transplant, there's a reasonably good chance that I've got a bit of your lung embedded in a paraffin wax block in the laboratory. And the reason we do that is that will allow us to cut tiny, tiny sections through the alveolus. So what we see, again, this is a cartoon, but I'll show you a real one in a minute. So what we see now is the actual cells that make up the alveolus. So there are different types of cells there. I'm going to talk about one in particular. But what a normal bit of lung would look like is this. It looks like a beautiful, almost like lace. The walls are very thin. The gases can pass very easily through these very thin walls. And although you can't see them unless you zoom in even further, within those pink lines, which are the walls of the alveolus, there are cells, and there are cells working really hard to maintain this structure and keep it intact so that your lungs function really well. When we look at samples from patients with LAM, what we see is this. This is what we call a cyst. Now, clearly the structure of the lung has broken down. It makes the lung less efficient. This is actually quite a small cyst. It's only about 1.5 millimeters across. And as well as the lung, I don't know if you can see right at the top. I'm sorry, I haven't got a pointer. There's a little bit of tissue sticking out. Actually, maybe my pointer finger will do it. It's up there. Those are the LAM cells. And those, we think, are what's causing the whole breakdown that forms this cyst. But the problem is, what we have here is a photograph. It's a snapshot of something that's already happened. And there are occasions when it's clear that something bad has happened, but it's not entirely clear how it happened. So this is what we're working with when we work with these tissue sections of lung. And so what we have to do is we have to identify the features that are unusual. So we see cysts and we see LAM cells, and we say, how did this happen? But you know, it's not obvious. We have to think about what might have happened. And so we look at the unusual features. Now we look, in the laboratory, we look at all the unusual features about LAM. We look at the cysts, we look at the cells that make up the nodule, but today I'm just going to talk about one particular cell. And I don't know if you can see, but in these slightly disorganized LAM nodules, this is a collection of LAM cells that we call a nodule, there's a layer of cells just around the outside, and here's some here, and there are some in that section over there, and some all the way around the circular bit there, and around the top circular bit. There's just a little, very organized looking layer of cells. We know what these are. These are type 2 alveolar epithelial cells. Now these are really, really important cells for the lung, because what their job is if other cells in the lung wear out, it's their job to replace them. So they divide and make you a new cell every time one of your old cells wears out. And normally they're dispersed throughout the lungs. They're just scattered there. There's something weird about this. They're not supposed to be all together in one place, lined up like that. That is a very unusual occurrence for these cells. There's something odd going on, and it is going on because they're next to the LAM nodule. The LAM nodule, the LAM cells are somehow influencing these cells and making them form a coating all over the nodule. And not only are they in the wrong place and too close together, they're also acting in a slightly hyper way, and we call this an activated phenotype. They're expressing proteins that they don't normally express. And they're not any old proteins. These are proteins that actually can influence the LAM nodule itself. They're things like growth factors, FGF7, that can make cells grow. MMP7 is a protease that breaks the matrix down. So these cells, not only now are they around the LAM nodule, they're behaving in a slightly odd way. They're also, we know, dividing a lot more quickly than they normally do, which maybe is why there are so many of them there. So we can stain the cells, these cells, for a particular protein that's only expressed in them, and that's what the blue stain is. And then we can stain them brown if they're dividing. And what you can see is around these LAM nodules, there are quite a few brown cells. If I look in the rest of the lung, I don't really see any other brown cells. So now we have an unusual feature to look at, which is the distribution of these cells. How might it have happened? We have to generate ideas as to how it might happen, and we call these our hypotheses. And what we do then is we go into the lab, and on the bench with cells in culture, we have to test the ideas that we have and see if we can make things happen on the bench with cells in a dish that are happening in the lung, so that then we can really drill down into the mechanisms of what's happening. Because clearly we can't do that in the patients. We have to rebuild LAM in a dish. So this is our idea, it's our hypothesis. And it is that LAM fibroblasts, which is a type of cell that's actually in that nodule, they attract those cells, which are called epithelial cells, and they make them grow. So there are two things going on. One, they cause them all to come towards the LAM nodule. And the second thing is they make them proliferate or grow. Why is this bad? It has to be something bad in order really for us to think about addressing it in a research way. Well, we know that these cells are producing things that could affect the growth of the nodule. We know that because we've looked. It could be that these cells actually are the cells that are in the nodule, but they change their shape, they change their phenotype, they change the way they look, and actually then they contribute to the bulk of the tumour. So it might be actually that those cells are a lot of the cells of the nodule. And the other thing is, if you have a lot of these cells around the nodule, but they're supposed to be somewhere else, and their job is to repair the lung, then if they're not repairing the lung somewhere else, then maybe the lung just becomes more fragile. Maybe that's the problem, that all the cells are drawn towards the nodule. So this is what we've been testing. If you've had a biopsy or a lung transplant, we may have done this with your piece of lung. We'll have taken it and we'll have dissociated it in a test tube and generated lots of cells, which will have grown in culture in a flask. Now, if those LAM cells are releasing a signal to cause these epithelial cells to misbehave, they're now releasing that signal into the flask, and we can collect it. So we take that medium and we put it onto epithelial cells, and we say, well, does it make them grow? And in fact, it does. So if we put epithelial cells just in plain medium, they're perfectly happy. They will sit there. They won't do anything. They won't proliferate. They won't grow, but they'll be quite happy. If we want them to grow, we add a bit of serum, and then you can see that this bar is higher than this bar because the cells are growing. But if instead of serum we put LAF, LAM fibroblast, conditioned medium that the cells have grown in, they grow. They really do grow. So actually, it's not quite as good as serum, but it's pretty good at making the cells grow. So we know now that LAM cells produce something that make epithelial cells grow. But can it make them move? That's the thing. Can it draw them towards the nodule? This is a different kind of assay. We call this a migration assay. The epithelial cells are in the top part of this well. And they're sitting on a little platform that has holes in it. They could go through the holes if they wanted. The holes are pretty small, so they have to put a bit of effort in to go through. And if you just put plain medium in the bottom, they don't make the effort and they don't go through. But if you put medium that's had LAM fibroblasts growing in there, they shoot through those holes. They really do migrate through those holes. So they like something about that medium, and that medium attracts them through. So that's quite good. We've shown the two parts of our hypothesis, our idea. One, it makes the cells grow. Two, it makes the cells migrate. It's nice to use a different kind of assay. So this assay is called a wound healing assay. We take a whole layer of epithelial cells and then make a big scratch in it. It's called a wound. It's just like a wound. And the cells, actually, they'll go, oh, for God's sake, and then they'll start healing. They'll start migrating, and they will fill in the gap. And they'll do that quite slowly. Even in plain medium, they will migrate. But if you put LAF conditioned medium that's been in contact with LAM cells in there, they migrate much faster. So again, it's a different way to show that LAM fibroblasts can make epithelial cells move. So that's good. That's the two parts of our hypothesis. But all of these experiments were done with cells growing on flat plates, and LAM nodules aren't flat. And what we always want to do is make the absolute best model in a dish that we can. So to make a better model, what we can do is grow the cells in 3D. So instead of growing the cells on a flat plate, we grow them as a sphere. We actually let them clump together, and we make something that's a bit more like a nodule. Now, we colour the epithelial cells green and put them in. What happens? Well, they love the clumps of cells. They love the LAM spheroids, and they will stick to them quite happily. And they will also start to proliferate, so we have more cells. So now we've made our assay a bit better, because now what we've got is something that looks a bit more like LAM. We have these fibroblasts, our LAM nodule, and we have the epithelial cells around the outside. It's getting to look more and more like LAM does in real life. But can we make it better? And we can. If you've had a lung transplant or a biopsy, and we've taken some of your tissue into the lab, we might have made these from it. Arudongre, who was a PhD student funded by LAM Action, called these lamospheres. What they are little clumps of tissue, not individual cells, they're little clumps of tissue that have come from the LAM lung. And we think they represent the nodules directly. We think what we have here are the actual LAM nodules growing in a dish. We know they have fibroblasts in, and we know they have LAM cells in them. So now these are absolutely the best model of LAM that we can grow in a dish. But we can only get these if we have a fresh lung to generate them from. So now let's make our assay a little bit more difficult. What we want to do is say, are the epithelial cells attracted to these, and will they stick? But we make it harder. Instead of letting them fall under gravity, we make them work against gravity to get to these lamospheres. So it's a similar assay to before. So these here, these are the lamospheres in a dish. They're growing in the same type of dish that I told you about before. They're sitting on a platform that has holes in it. But now the epithelial cells are underneath. If they want to get to the lamospheres, they have to go up against gravity. And that is exactly what they do. They will climb through the pores in the platform, and they will stick, here they're coloured green, to the lamospheres. So now this is our absolute best model of this interaction between two types of cells, two epithelial cells and the LAM cells, that we can build. So what we have so far is, our hypothesis was that LAM fibroblasts attract epithelial cells and cause them to proliferate. We can show that both of these things are true. And this could account for what we see when we look at the patient samples. We could account for the fact that LAM nodules are often coated by this layer of epithelial cells, and that these are more proliferative. And now we have this model. What we can start to do is work out what the mechanism is. And there are ways we can do that, because there are already identified pathways from other diseases that we can stop working. And so we stop a pathway working, and we say, does it affect this process? And it may affect it, in which case we can say it's involved, or it might not affect it at all, in which case it isn't. But because these are very well understood and very well established pathways, there are lots of drugs that actually we can use to stop them working. So that is that part of the work. But on a similar vein, we're also looking at how the cysts form at the very, very earliest stages. So cysts that are from a fraction of a millimeter to a few millimeters in size. we can look at them in our patient samples in the sections, and we can have a look at what types of cells are there at that point. And that's one of the things we're doing. We can say what cells are there and how bad is that patient's LAM, and how does that disease progress if it has this cell, or if it has epithelial cells or no epithelial cells. And it seems clear from these early cysts that actually at this point they don't have the epithelial cells. They seem to come in later. So they may contribute to the later stage of the disease. And also, we look at the later stage of the disease where actually it's a bleb on the pleural surface, but you can see there are lots of different things going on in there, some in just a subset of cells, some in all the cells. And we can work out how all of these different proteins might affect that patient's disease. And in some cases, we can see evidence of the disease actually before we can see a cyst. So we see at the top, you can see the slightly darker brown part there. That's a marker called smooth muscle actin, and there are other markers there that are characteristic of LAM. So it looks like there's a little patch of LAM here, but there's no cyst associated with it, presumably yet. So the cyst might be the next step along. So that's just a snapshot of one part of the research that we're doing. But as you can see, that we use the patient samples really to design our experiments. We gain our ideas about what's happening in LAM from the LAM patient samples. And then we use the LAM patient samples to do our experiment. And then what we will do is we'll identify all these pathways and factors in the lab, and then we will go back to the patient samples, and we will make sure that the factors we've identified really are active in LAM. So that's the way that our work is progressing. And I should say, thank you to LAM Action, who've funded my work, my salary for, I think, 16 years. Both Suzanne and Roya have contributed to this work. Roya makes the spheroids. Her work is funded by the TSA, and she's interested in how the immune system affects LAM. Suzanne's work is funded by the LAM Foundation, and she's interested in how the cysts form and whether proteases are involved in that. But I should say that the funding that both of these postdoctoral researchers have was acquired on the basis of research that was done and funded by LAM Action. So actually, the LAM Action grants that we've had have attracted funding from other sources. So thank you.